Minerva and I'm working in Dr. Ramos's lab in the Medical Research Building. The technique I'm going to be demonstrating today is SDS page. Does forcing the expression of line 1 proteins in non-malignant cells transform these cells to cancer cells? We begin by taking culture lung cells out of the incubator. We are going to demonstrate the lysing the cells to extract protein, preparing the samples and loading the gel. The first step is to remove the media from the cells. We then wash the cells with ice-cold PBS. Next, we prepare the lysing reagent and the cells are processed for lysing. Once the cells are lysed, we then use a scraper to scrape the cells around the container and set them to the bottom. This step is to assure we collect as many cells as possible. We extract the cells from the flask and transfer them into a test tube to further prepare our samples. Our test tubes are centrifuged for 10 minutes to get the samples ready for measuring their protein concentration. We then mix our BCA protein assay kit. We then begin loading the samples in by triplicates and proceed to add protein assay reagent to all the wells containing the samples. The well plate is then loaded onto the Citation 3 imaging reader where the protein concentration is measured and recorded. After protein concentration is measured, samples are prepared in a new tube with loading buffer and heated to 90 degrees Celsius for 5 minutes. A pre-made glycine gel is inserted into the tank with running buffer. The gel is loaded with a protein ladder and samples are loaded into the wells. An electrical current is connected to the tank and set to run for 1 hour. SDS page is a technique used to separate a protein mixture by size through gel electrophoresis. My project was to screen BEAS2B cell clones expressing line 1 and mutant line 1. My objective is to force expression of line 1 proteins in non-malignant cells to test if line 1 protein transforms cells to malignant cells. This technique helped us to answer the question whether the cell clones generated express line 1 proteins.